This is our project on methamphetamine, informally known as crystal meth. We'll explore the way it alters cell communication, leading to lasting damage in the brain and body. By Eowyn and Vivian.
First up are the ligands involved in this process. The ligands include methamphetamine, which is its own ligand, displayed here, or neurotransmitters, such as dopamine. Dopamine, which is naturally in the body, is produced in multiple brain areas, including the substantia nigra, which is located in the center of the brain.
So what does meth do? Displayed are the presynaptic and postsynaptic areas. At display now are dopamine transporters in blue, or DATs. This is the usual process of dopamine release in which dopamine molecules are stored in vesicles to be released at periodic intervals. However, this process is disrupted when meth is introduced. As meth enters the presynaptic area, it alters the function of the protein VMAT2, which is responsible for vesicle formation. The dopamine inside the synaptic areas is no longer constricted by vesicles and moves freely through the DAT and binding freely to the dopamine receptors. This induces an influx of dopamine release into the synapses as methamphetamine repurposes the dopamine transporters to excessively pump out dopamine. Furthermore, the meth binds to the DAT and alters its function so the uptake of dopamine through the dopamine transporters is blocked.
So let's elaborate. Meth can enter through the DATs due to their similarities in composition to dopamine and can also pass through the membrane through passive transport. The effects of meth lead to large dopamine increase, as mentioned previously, and the excess dopamine stuck in the synapse then transforms into quinone or semiquinone. These compounds catalyze the production of harmful byproducts known as reactive oxygen species, such as hydrogen peroxide, OH negative, or O2 negative. These are oxidative stress reactions, such as lipid peroxidation, which triggers cell death or damage.
Next, we look at our messengers in the signal transduction pathway. In this cell signaling process, when dopamine activates the alpha and beta receptors, they activate GPCR proteins, which interact with the adenyl cyclase, displayed in the model on the left. The excess dopamine ligand then interacts with the receptors on the left. The presence of the ligand causes the receptor to send signals to the adenyl cyclase, which in turn transforms ATP into the secondary messenger CAMP. The CAMP then attaches to protein kinase A, which is shown on the bottom right. This process then leads to consequential cellular responses, in this case, neuronal excitability. To recap, the first messenger in the pathway are the ligands, methamphetamine, and dopamine. The second messenger is the CAMP.
Overall, the results of meth are 1. Dopamine spikes, 2. Excess dopamine, which leads to harmful byproducts such as the reactive oxygen species, and eventually start a cell death cascade. 3. It hijacks cell transduction pathways and increases neuronal activity. Now let's look at the effects of methamphetamine on the body. Starting off on the brain, methamphetamine causes euphoria, hyperalertness, decreased fatigue, better focus, anxiety, and reduced appetite. Next is the heart, where results include an increased heart rate or vasoconstriction. As for the lungs, increased breathing rate can be a consequence. In the immune system, function is impaired. As for the muscles, tremors, muscle tension, or spasms.
Next, we have the long-term effects on the body. Starting off on the brain, memory deficits, psychosis, depression, and neuron damage are all long-term results. For the heart damage, arrhythmias, atherosclerosis, and increased risk of stroke. The lungs can suffer permanent damage. The immune system is at an increased risk of infection, and methamphetamine use weakens and suppresses immunity. The muscle can suffer from tissue damage and harmful protein release. Lastly, the reproductive system can be affected in terms of reduced fertility, erectile dysfunction, and menstrual irregularities.
This concludes our project. Thank you, and say no to meth.